We will start with question number 1. Which of the following cuboidal cells would have the highest efficiency with each of its sides measuring 2 mm, 20 mm, 200 mm, 2000 mm?
2 mm.
Okay, anyone else?
2 mm, sir.
Okay,
Sir,
so
2 mm.
okay, so all of you are with 2 mm, that is the smallest size. Okay,
Yes, sir.
so the smaller the size of the cell, okay, smaller the size of the cell, higher the efficiency. So
Yes, sir.
size of the cell and efficiency
Inversely proportional.
inversely proportional to each other. Why is it so? Because when the size
So
of the
because,
cell,
so because small size has higher surface area, which provides greater area for the
yes,
action of enzymes and
yes,
so provides higher.
exactly.
Volume to surface area ratio is more in that case.
Yes. So if you have one cube, that is with this 2000 mm cube, okay, 2000 mm cube is there. In that one, how many 2 mm cubes can come inside? 1000 mm
1000.
cubes can come inside one
Yes,
point.
sir. 1000.
So they will have the four sides. Okay, the four all the sides which are there, they can all absorb the nutrients. So they will have a higher surface area. They will have higher efficiency. So that is a good point. The surface to volume ratio is high in the case of small cells. Okay, good. Next point. A combination of ordinary light and glass lenses is used in the simple and compound microscopes to magnify an image. What is used in the electron microscope?
So D.
So the electron beam
Electron
and
beam and
magnets.
magnets.
Correct? Electron beam and magnets. So whereas the ordinary microscope, we are using the ordinary light, that is the white light, what we say, and we are using the glass lenses. Okay, so those glass lenses, they will be concave or convex?
Convex. Convex.
Convex, right?
Convex.
Convex. If it is a concave, sorry, if it is concave, it will be something like this. Okay, but this is the convex. So the convex lenses will be used, and a mirror will be used, that is a concave mirror. A concave mirror may be used to, what you say, colliminate the light, that is to concentrate the light near the sample. Okay, so
Yes,
but
sir.
in an electron microscope we are using electron beam. We are using electron beam instead of the ordinary light. So what is the difference? Is this wavelength of the electron beam more than the ordinary light or less than
Less than.
lambda
Less.
of ordinary light,
Less.
white light and electron beam? Which is more?
So electron beam will have more wavelength.
More wavelength. Okay, anyone else? More wavelength means, let us say, longer wavelength. Okay. Longer wavelength.
So lambda value will be high, I think.
So you see, if you can also look in this way, that wavelength is inversely proportional to frequency. Okay, so here if the frequency is high, that means energy will be
More.
more, right? Because
More energy.
yeah,
More
frequency
energy.
and energy are directly proportional to each other. So here when we are telling a electron beam, the electron beam, we want to look very fine, no? Sample we want to see between very close structures. So we need a wavelength that is, should be so small. Okay, if it is having a long, then what is going to happen is that its ability to distinguish between these 2 points, close points, will be less. Okay, so here important thing is that electron beam is a high energy beam compared to the white light. Okay, high energy beam, it has a higher frequency, lower wavelength, low. short wavelength. Okay, electron beam is a short wavelength. Anyway, so these magnets, what they are going to do, because this is electrons, no, they are having negative charge. So these magnets can be used to shape the beam. Okay, next. The nature of magnifying glass lens and light collecting mirror used in compound microscope are respectively.
Sir, option C.
Option C. By convection control.
Correct. Other than the nucleus, DNA may be present in the cell in.
So mitochondria.
Mitochondria.
All of the above.
All of the above. Okay. Mitochondria also, chloroplast also, and in the prokaryotes it can be present in the plasmids. Okay. The chloroplast also has its own DNA. Okay. So these two things, that is the mitochondria in the case of eukaryotes, chloroplast particularly in the plants, and these plasmids in the prokaryotes, they all have the DNA in addition to the regular nucleus of the eukaryotic cell. Genes are made up of, this is a direct easy.
RNA. RNA.
Genes are made up of RNA. Okay. Anyone else?
Sir, DNA.
Sir, genes are made up of DNA.
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 The DNA.
So what is the thing is, the DNA is the storage material of the all the genetic information. Okay.
Yes, sir.
There are exceptions. For example, coronavirus is an exception.
Yes, sir. It's an RNA strain
It
virus.
is RNA. Okay. For its
Yes, sir.
the genetic material is RNA. Okay, that is exception.
Yes, sir.
So, but the general trend when you look at, so DNA is the genetic material for most of the organisms because DNA is very stable chemically compared to the RNA. What happens is this DNA will be converted into RNA, and this RNA will be converted into protein. Okay, it will have some three-dimensional shape that can do some function. Okay, so within this DNA we have short segments which can function as genes. Okay, they will code for some specific protein or do some specific function. Okay, so that is one thing.
Sir,
So
this in RT-PCR test is also used that RNA is converted to DNA. So in
in
RT-PCR test,
RT-PCR what happens is this reverse takes place. RT
so
means
reverse
what?
transcription.
Transcription. Okay, this will convert back into DNA. RNA will convert into, so specific enzyme is there that can do that. Okay, so I